There we go. Alright, so it's 9 p.m. on the night before the deadline. Today we're going to go over cell communication, and so all the receptors, secondary messengers, all that good jazz. I think to get a true understanding of cell communications, you got to first know the 3 basic steps that goes into everything that occurs in the cell. The first step is reception. This is the detection stage of cell communication. It's where the outside signal comes in and kind of starts it all. The next step is transduction. This is all the intermediate steps that go in before the actual cellular response comes out. So all the intermediate steps that has to occur. And last but not least is response. Is that what it's called? Yeah. It's the response stage. This is when the cell actually does something, and so it's the ending result. So basically, reception is it receives a signal and kind of starts the whole reaction. 2, transduction is everything in between that leads up to the cellular response, and 3 is the actual result of the signal transductions and all that. Now that we know the basics of cell communications, let's move on to the the pathways. Alright, so we're going to start with the G protein pathway. This is the most common pathway, and it deals with stuff like your vision, smelling stuff, tasting. So it's pretty important. The picture you're looking at right now, nothing's really going on because it's largely inactive because there's nothing there yet. But what occurs is a ligand. This is the signaling molecule we were talking about earlier. Once it attaches to the receptor, it changes the shape of the receptor, which brings it from an inactive to an active state, and that causes this G protein to shift on over and attach to it. So once the G protein, once it attaches to the receptor, the GDP that was here, it's possible to get phosphorylized to GTP because of the shape change of the receptor, and that makes the G protein active, whereas when it had GDP it was inactive. So after that, the G protein, it diffuses across the cell membrane and acts as a relay messenger of sorts, because it carries the next message for the transduction processes, and it attaches to the enzyme, which the shape of the enzyme changes, so therefore the structure of it, the structure defines the function. Alrighty, so with that, the enzyme, it can carry on whatever the rest of the processes of the G protein pathway, and it leads to the cellular responses we're ultimately looking for, and that is the G protein pathway. Another thing about it is if the ligand were to, leave the G protein, it stop binding to the enzyme, it revert back to its inactive stage, and then the ATP that was here, it becomes hydrolyzed to ADP and phosphorus, so then it reverts back to ADP here, and that stops the reaction. So next we have the tyrosine kinase pathway. This pathway, it deals with cell repair and all that. And the receptor consists of 2 monomers, and each have their own individual ligand receiving sites. And each monomer has 3 tails of tyrosine kinases. And what occurs is once ligands attach to the sites, the monomers undergo dimerization, and pretty much what occurs is they become one and form a complex. And once this complex is formed, the tyrosine kinase tails, they become activated, and this causes ATP around in the cytoplasm to give off 6 ATP to all give off one phosphate to each. The ATP, once it gives off, the phosphorus becomes ADP. So then once all the tyrosine kinases are activated, relay messengers within the cell, they come and bind to all 6 of the tyrosine kinase tails. Let me just draw real quick. And this changes the structure of each of the proteins that come, and because of that, their structure defines their function, just like every other pathway we're going to go through. And this causes them to move on to the next stage and perform. They just keep going on and on until a cell response occurs. So basically, right here we have the reception and transductions, everything in between here, where the proteins come and receive, and the cell responses is down the road. Next, we're going to deal with ion channel receptors, aka ligand-gated ion channels. These allow/block certain signals to enter or exit the cell even. And when it says ligand-gated, it's not always true, because some of these receptors use electrical signals to determine whether they open or close. And so what occurs is, once a ligand enters the site, the receptor, it opens up, and this allows for the flow of all, or for this one we're using sodium ions, so they all flow into the cell cytoplasm, and this causes a concentration change within the cell, and that can sometimes automatically lead to cellular responses. And that's about it. But then when the ligand is removed, the receptor closes back up, and no more of the sodium ions can flow back in. Now we got intracellular receptors. These deal with lipids, so like hormones and steroids. Since these things are hydrophobic, they don't need any specific receptor or anything on the plasma membrane for it to enter the cell. What occurs is, we're going to use aldosterone, it's a hormone, as our example. What occurs is they simply diffuse through, and once they diffuse through, there are intracellular receptors waiting for them. And these receptors, once the hormone goes in, it binds to the intracellular receptor. And the reason it's called intracellular is just because it's within the cell, rather than like on the plasma membrane. So once the hormone attaches to the receptor, the shape changes, and it forms a complex, and this complex moves towards the nucleus. And in the nucleus, there's specific genes, so DNA. What's it called? Intracellular receptor. It acts as a transcription factor because it transcribes DNA. It transcribes mRNA from the DNA. So it pretty much activates and starts it, and it forms mRNA. The mRNA, after it's transcribed, it moves out of the nucleus and goes to the ribosomes to be made into a specific protein. But that's outside the scope of this video, so we'll be moving on. Alright, now we're moving on to secondary messengers. All secondary messengers is pretty much they're relay messengers within the cell that move around in the cytoplasm. They pretty much just carry through all the different stages of transduction. And so let's assume we've already been through the G protein pathway. So ligand. Oh yeah, the blue marker stopped working, so we're really limited in colors. What happens is, okay, so let's assume the ligand's already here. The G protein gets activated, GTP, and the G protein moves to the enzyme and changes its structure. So with this enzyme is adenyl cyclase. That once it's activated, it turns ATP into cyclase AMP. Yeah, cyclic AMP. So CAMP for short. And what this does, it acts as a secondary messenger that continues the process of transduction. And what it usually does is it goes ahead, moves forward, and protein kinase A, it activates that, and since this is a kinase, it goes on and phosphorylizes other parts of the processes, and then that eventually leads to cell responses. Alright, my phone's only at 4%, but we are at last video. Secondary messengers, calmodulin, and IP3. These deal with muscle contraction, according to the outline. So, again, we're assuming the G protein pathway has undergone, but this can also go through tyrosine kinase. So ligand changes shape and activates the receptor. The receptor attracts the G protein, phosphorylized by ATP. So this becomes GTP. G protein moves on. And this time we have PIP2 that was previously the inactive enzyme. But what's slightly different here is instead of just binding, for some reason we decide to cleave PIP2 and straight up chop it up, and it becomes IP3 and DAG. DAG is another secondary messenger, but it has nothing to do with, well, it just goes on and does to different pathways and through transduction, but that's not a part of what we're doing. The IP3 as a secondary messenger. So the cell has an endoplasmic reticulum. The IP3 moves to the endoplasmic reticulum. The ER, it has a receptor here. That's a gate because the ER is filled with calcium ions. So this gate, IP3, will serve as a ligand to open up this gate for the calcium ions to all move out. And then once the calcium ions are out, they move to the microfilaments of the cell, causing your muscles to contract. And the example of muscle contraction is a cellular response, which is what this is all about. And so I hope you enjoyed the presentation. I hope to never do this again, but thank you guys for watching, and bye-bye.